What's demyelination? Demyelination is the loss or damaging of myelin in nerve cells. So, what's myelin and what's a nerve cell? Nerve cells are cells of the brain. They transfer signals like wires conducting bioelectric signals. These cells can either be myelinated or unmyelinated. When a nerve cell or neuron is myelinated, its axon or stem is wrapped with a fatty tissue called myelin. This myelin insulates the nerve and allows the nerve to send messages to the rest of the body at a faster rate. Myelin is mostly found in the neurons of vertebrates, but also in a few worms and crustaceans. And nerve cells are present throughout the entire body. Pretty cool. In certain diseases like multiple sclerosis, MS, or Guillain-Barré syndrome, GBS, the body's own immune system may directly attack the myelin of nerves. It's important to note, though, that Guillain-Barré syndrome and multiple sclerosis are totally different diseases. GBS occurs in the peripheral nervous system, or PNS, where multiple sclerosis occurs in the central nervous system, or CNS. So what's the big difference? We interviewed Professor Goldman of University of Rochester to find out.
So the Schwann cell is different from the oligodendrocyte. They're actually derived from different cell types embryologically. So effectively, they've converged from the evolutionary standpoint to fulfill the same function, which is insulating long nerves, but from two different cell types in ways that are very different. So the proteins that are made by oligodendrocytes and Schwann cells are actually different.
Okay.
So you can get autoimmune destruction of peripheral nerve myelin as well. There are lots of autoimmune conditions that target peripheral nerve myelin.
So, oligodendrocytes and Schwann cells. What's the big difference?
Disorders, the oligodendrocytes and the Schwann cells are very different. They're also very different in terms of how they behave biologically. So that oligodendrocytes, one oligodendrocyte will wrap upwards and sometimes of 100 axons. So the individual nerve fibers, one oligodendrocyte will unsheath many, many axons. A Schwann cell just unwraps one axon. That's it. And so that's a really important difference between the two. So you can just imagine that if you lose a Schwann cell, it's not necessarily that much of a loss because you're losing basically the unsheathment of one segment of axon that's right next to it.
Alright, so oligodendrocytes make myelin in the brain, whereas Schwann cells make myelin in the body. MS is a disease that attacks the CNS myelin in the brain, whereas GBS attacks the peripheral myelin in the body. Which damage is worse?
When you lose an oligodendrocyte, you have anywhere from, depending upon the region of the brain, 20 to 100 axons that are going to be demyelinated because of it. So loss of an oligodendrocyte can be much more damaging. Oligodendrocytes also can't divide. They're just like neurons, they don't divide. New ones can form from what we call glial progenitor cells. Some people call them oligodendrocyte progenitor cells, but it's actually a type of an early progenitor, or some might even say a stem cell in the brain.
Peripheral myelin regenerates almost completely, as seen in recovering GBS patients. But why doesn't this happen with the myelin in the CNS?
You can remyelinate a demyelinated lesion in the brain slowly from the oligodendrocyte progenitors making new oligodendrocytes. But that doesn't happen very efficiently, and as a person gets older, it happens less and less. And so by the time somebody is elderly, it's not clear that it happens at all.
So, how are demyelinating diseases being treated?
Kinds of numbers of human oligodendrocyte progenitors that will be necessary for clinical treatment.
Professor Goldman mentioned the use of progenitor cells in these treatments. Progenitor cells are stem cells. Stem cells differentiate into specific cell types, like oligodendrocytes or Schwann cells. Scientists are just beginning to understand the signals inside and outside of cells that trigger this differentiation process. Learning to control differentiation of stem cells by characterizing all of the signals is a critical science. Complete knowledge of this helps develop stem cell treatments for demyelinating diseases.
So now that this treatment is known, what's standing in the way of making MS a thing of the past? We interviewed President Jamshed Bharucha of the Cooper Union, experienced neuroscientist, to find out.
Well, I would say the biggest obstacle is the difficulty in translating research from the basic sciences into clinical treatment. And that has been recognized by the NIH, the National Institutes of Health, for some years now, which is why the NIH has been pushing for translational research, which, as its name suggests, taking research from the lab to the...
So there you have it. The science is known and the theory is out there. Now, the physical treatments begin as we wait for what the future holds.